Thank you so much, Jason. Thank you to the organising team. It's a real pleasure to be involved in this really exciting symposium. I've enjoyed the talk so far very much. Yeah, I'm here to tell you a little bit about our genetic studies of birth weight. So over the last sort of 25 or so years, studies of a variety of populations have documented a really interesting association between type 2 diabetes and birth weight, such that people born in the lowest categories of birth weight are more at risk 50 or 60 years later of developing type 2 diabetes. And the underlying reasons for this are still not fully understood. So the observation originally led to the fetal origins hypothesis proposed by David Barker, under which a poor intrauterine nutrition would be expected to lead directly to a smaller, thinner baby, but indirectly via programming in utero, effects of the in utero environment on the developing child would be expected to lead to an increased risk of type 2 diabetes. And there's good evidence from animal models showing this kind of programming effect. And in humans, the best evidence is from studies of extreme famine. But another explanation for the association between lower birth weight and type 2 diabetes was proposed by Andrew Hattersley, which is the fetal insulin hypothesis. And so under this hypothesis, a lower birth weight and a higher risk of type 2 diabetes are two phenotypes of the same genotype, because genetic variants that influence the ability to either produce or respond to insulin would be expected to influence risk of type 2 diabetes. But since fetal insulin is a key fetal growth factor, those same genetic variants might be expected to influence fetal growth. So the evidence on which the fetal insulin hypothesis was originally based came from studies of mothers and children with glucokinase mutations. So the glucokinase mutations, inheritance of glucokinase mutations cause the fetus to produce less insulin, leading to reduced insulin-mediated fetal growth. Now, in children and adults, those same mutations cause a mild, stable, fasting hyperglycemia, so higher fasting glucose, again, due to that reduced ability to produce insulin. So what Andrew and colleagues found was that in sibling pairs who were discordant for glucokinase mutations, those siblings who had the mutation were on average half a kilogram lighter at birth than siblings without the mutation. And this was really good evidence that fetal insulin was a key fetal growth factor, and this is a clear genetic effect linking lower birth weight with diabetes later in life. Now, the other thing that the glucokinase example really illustrates nicely is that both maternal and fetal genes can influence birth weight independently. So in this is data considering now not just mutations in the child, but also in the mother. So when the mother has the mutation, but the baby does not, then the baby is born with a higher birth weight due to increased fetal insulin secretion in response to a higher level of maternal glucose. When the child has the mutation, but the mother does not, then the baby is born with a lower birth weight due to a reduced insulin secretion in response to a normal level of maternal glucose. But when both mother and child have the mutation, those two opposing effects cancel each other out, and the baby is born with a birth weight that's no different from average. So just to summarize, what happens is that maternal glucose crosses the placenta, a fetal glucokinase mutation reduces glucose sensing by the fetal pancreas, reducing insulin secretion and insulin-mediated growth, reducing birth weight. But we see this opposing effect in the mother, whereby the glucokinase mutation reduces her own ability to produce insulin, leading to more availability of glucose, higher fasting glucose, which crosses the placenta, causing increased glucose sensing by the fetus and increased insulin-mediated growth. So of course, those rare mutations established the principle of a genetic link between lower birth weight and diabetes, but of course they can't explain themselves explain the association observed in general populations because they're so rare. So our efforts to sort of try and understand this have focused on studies of common variants. And over the last sort of 10 or so years, we've been performing genome-wide association studies of birth weight that have got successively larger. And in our most recent study, we were able to look at a very large sample of individuals who have their own genotype and information on their own birth weight, which we call the fetal GWAS, just over 320,000 individuals in that, from studies in the EGG Consortium and the UK Biobank. And then we also were able to look at a sample of 230,000 mothers with information on their own genotype and their offspring, but birth weight of one offspring. Now, the sort of results of that are shown on this kind of mirrored Manhattan plot, where we saw we identified a total of 190 genetic loci associated with birth weight. But what you can see is that there was clear overlap between the maternal and fetal signals. So 28 signals were identified in both the maternal and the fetal genome-wide association study. Now, of course, the mother and child's genotypes are correlated. A baby gets half its genes from its mother and half from its father. So when we do a fetal genome-wide association study of birth weight, we are capturing information about the mother's genotype and the father's genotype. And when we do a maternal genome-wide association study of offspring birth weight, we are capturing some information about the fetal genotype. So to really understand the results of our genome-wide association studies of birth weight, it's essential that we separate our estimates of our fetal effects from maternal effects. And as was really nicely illustrated by the glucokinase example, a maternal gene can be passed to, maternal genetic variant can be passed to the child and directly influence birth weight. But regardless of whether they're passed on to the child or not, maternal genes can also influence birth weight indirectly via an effect on the in utero environment. So we were really keen to separate direct effects of the fetal genotype from indirect effects of the maternal genotype. Now, to do this, ideally, we would need a sample of mothers, fathers, children, that was at least as big as our discovery genome-wide association study, so that we could, for each locus, do a conditional analysis and say whether it was a fetal or a maternal effect. But of course, that data is really hard to come by. We need, we just simply don't have those resources available. Currently within the EGG Consortium, we have about 30,000 mother-child pairs or parent-offspring trios where we can do this kind of analysis, but that's not enough to get the kind of granularity that we need to look at the individual loci. So that's where a method developed by Nicole Warrington and Dave Evans in Queensland has been wonderful and has transformed what we can do with these studies. Because what Dave and Nicole did was to use the sort of unique structure of the UK Biobank, where participants have information on their own birth weight, and also some of them have information on their offspring birth weight, to fit a structural equation model to actually jointly estimate maternal and fetal effects. And this does really well compared with using sort of mother-child pair data. And so so we were able to apply this to the results of the maternal and fetal GWAS. And so this slide just illustrates what can be done. So on the left-hand graph are the loci that were identified in the fetal GWAS, so the GWAS of own birth weight. And what's plotted is the estimated fetal effect using the structural equation model. So that conditional on the maternal effect, and similarly the maternal effect taking into account the correlation with the fetal effect. And for these loci that were identified in the fetal GWAS, as you can see, there's a few here that have zero fetal effect. These grey lines are sort of zero lines on each axis. So there's a few here that have zero fetal effect, but an appreciable maternal effect. So these were picked up in the fetal GWAS simply due to their correlation with the maternal genotype. And similarly, those loci that were identified in the maternal GWAS, there's a number here which have a zero maternal effect, but a fetal effect. This has been really useful in downstream analyses, trying to understand what these loci are doing a bit more. So going back to our sort of links with diabetes and glucose-related traits, there's a lot of information on this slide, but actually what I'd like you to do is just focus on what's in the red oval. These are genome-wide genetic correlations between our GWAS results for birth weight, maternal effects sort of adjusted for the correlation with the fetal genotype in triangles, and the circles are the fetal effects adjusted for their correlation with maternal effects. And what you can see is for type 2 diabetes, fasting glucose and other related traits, those where we see a higher risk or a higher level of glucose or higher risk of type 2 diabetes, the maternal associations with birth weight are generally positively genetically correlated, and the fetal have this negative correlation. So this is consistent with there being a genetic predisposition to reduced insulin, which is also showing as an effect on reduced birth weight, so supporting the fetal insulin hypothesis. So just sort of as a little aside, as you can see, what the genetic studies are doing are helping us to understand mechanisms. And so instead of sort of looking at this as a way to understand the relationship between birth weight and type 2 diabetes, we can also actually view this as a way to understand the role that fetal insulin might play in fetal growth, or in this case, placental growth. Now, at the time of the studies of glucokinase variants, the rare variants that cause glucokinase related hyperglycemia. Bev Shields and Andrew Hattersley and others looked at effects on placental weight, and as well as finding in sibling pairs who were discordant for the mutations, that the siblings with the mutation had a lower birth weight, they also had a lower placental weight. And so this was evidence that fetal insulin produced by the fetal pancreas plays a role either indirectly or directly in placental growth. And very recently, within the EGG consortium, we've been looking at genome-wide association studies of placental weight. We've put together about 50 to 60,000 fathers, mothers, children with GWAS and placental weights. And this is some of the follow-up work that Rob Beaumont's been doing, looking at Mendelian randomization, taking genetic variants associated with fasting glucose or insulin secretion or resistance as instruments to estimate the causal effect of those characteristics on placental weight, either when considering the fetal or the maternal genotype and adjusting for the correlation between them. And what we see is really, it corresponds very well with those rare mutation studies and with what we see for birth weight, in that the higher maternal glucose is causally associated with a higher placental weight, and we see the sort of opposite effects for the fetal characteristics, again consistent with a role for fetal insulin in placental growth. So back to the sort of question of type 2 diabetes and birth weight and a genetic link between them. Looking at the individual loci, we can ask the question, do all type 2 diabetes risk alleles reduce birth weight if inherited by the fetus? Is it a general feature? Well, when we look, we see that it's not a general feature. So this was data put together by Alice Hughes looking at a number of type 2 diabetes loci that have been well characterized and clustered according to their likely modes of action in terms of risk of type 2 diabetes. And what you can see is that these are the fetal effects on birth weight of these risk alleles. And those implicated in obesity or in liver function seem to show no association with birth weight, but those associated with beta cell function or a lipodystrophy-like phenotype, there are a number which show fetal effects on birth weight. And so what this suggests to us is that these are acting on fetal growth via insulin secretion or insulin resistance. But also what you can see from here is that even though, for example, in the beta cell cluster, we see associations with birth weight, that's definitely not the case for all of them. And actually TCF7L2 at the top here, which is well known to have a relatively large effect on type 2 diabetes risk. In the fetus, we see no association with birth weight. And so that suggests that its effect on insulin secretion is not present that early in the life course. So whereas, for example, for CDCAL1, we do see that is present. The other thing to point out is that we do have this one here. This isn't a mistake. ANC1 is this is the risk allele for type 2 diabetes, and it's associated with a higher birth weight. So again, they don't all associate with lower birth weight, and there's even one going in the other direction. And another question is, we can look at it the other way, and we can say, do all birth weight loci influence fetal insulin? So is it a general feature of those loci that influence birth weight? And very quickly, I can tell you that no, they don't. There's actually only a small fraction that are implicated in type 2 diabetes and glucose-related traits. We see, for example, many height-related genetic loci that influence birth weight, likely through an effect on skeletal growth. And actually, when looking at the associations between maternal glucose, which we know crosses the placenta, encourages fetal insulin secretion, when we look at the associations between maternal glucose and cord insulin or C-peptide, which is secreted in a 1-to-1 ratio with insulin, in the Exeter family study and the HAPO study, we see there's a clear association with insulin, but actually a fetal genetic score for birth weight is not associated with cord insulin or C-peptide. With enough power, I would predict that those few type 2 diabetes loci implicated in beta cell function should associate with cord insulin, but we simply don't have the power to test that. But what this has enabled us to do is to think about genetics as an independent factor, independent of maternal glucose in terms of its effect on birth weight. And so this data from Alice has, again in the Exeter family study, shows us that even in women in the highest tertile of fasting glucose, so at highest risk of having a large for gestational age baby, above the 90th percentile, those women who have the highest tertile of fetal genetic score, sorry, the fetuses with the highest tertile of genetic score in those pregnancies have sort of double the risk of being born large for gestational age compared with those in the lowest tertile of the genetic score. So this raises the possibility that we could actually use information about the genetics of the fetus as well as glucose in the mother to get better at predicting birth weight. Currently in the UK, we identify the risk of macrosomia, or having a large baby, using ultrasound scans and the estimated fetal weight statistic, or by screening for maternal glucose levels if certain criteria are met. So, for example, if a woman has a BMI above 30, or if she's had a previous large baby or previous or family history of gestational diabetes, that can trigger a need for a glucose test. But these methods are not perfect, and there are many large babies born to women when that wasn't suspected, and many women who have gestational diabetes, for example, have a baby who's not large. So Manika Holder in Exeter has been looking at whether we can improve the variance explained in birth weight when we add certain characteristics that capture the fetal genotype to maternal clinical features. And when we just add in maternal heights and paternal height, height is a very genetic trait, and just measuring the paternal height, for example, should capture something about the fetal height. We see that there is an improvement in the variance explained. And when we add a maternal and paternal genetic score to this, the variance explained in birth weight improves again. And just to sort of show you that the model with the effect sizes here, the change in corrected birth weight per one standard deviation change in each variable is presented here. And as you can see, the changes that are conferred by the maternal genetic score are comparable to those for fasting glucose, and similarly with the paternal, the fetal genetic score and the heights. So this raises the possibility that we could, in high-risk pregnancies affected by gestational diabetes, we could actually use information about the fetal genetics, even by just measuring heights, to improve prediction. So just to summarize, I hope to have told you that lower birth weight and type 2 diabetes can be two effects of the same genotype. It can tell us something about mechanisms of fetal and placental growth, but not all type 2 diabetes loci influence birth weight, and most birth weight loci do not influence fetal insulin. And the fact that they are independent of the glucose effects means that we can actually begin to investigate using these other factors alongside glucose in terms of predicting birth weight or improving prediction of birth weights. So our future efforts are going to focus on diverse ancestries, and that's an area we're really trying to get the samples in at the moment. We are currently doing a GWAS of umbilical cord insulin, and we are also improving the samples of our larger studies of parent-offspring transmission so that we can investigate these more closely. So just to finish, just by saying that all this work is the work of many, many people in large consortia, and to thank my team and my funders. Thank you very much.